Everybody's quiet now. All right. We might as well make a start. So thanks everybody for coming along to this ERI seminar. And it's my great pleasure to introduce Dr. Simon Stewart today, who's a freshwater ecologist at the Cawthron Institute in Nelson. Simon has actually completed his BSc and MSc at the University of Canterbury. And then he saw the light and came to Waikato University for his PhD, which he completed in 2018. I think I've known Simon for much longer than that. We were talking about this earlier. I think we met in 2010 over beer and karaoke, but sounds a bit, but I don't have any recollection of that evening. So Simon works on a range of broad range of freshwater projects, often using stable isotope techniques in conjunction with a range of methods to address challenges pertaining to lake management and fishery management. These projects also include a mast and fast start that you are leading, Simon, that examines the role of inanga as a marine subsidy for supporting long-fin eel growth. Simon has also received the 2023 New Zealand Freshwater Sciences Society Early Career Researcher Medal as a recognition of the quality of his research. So thanks, Simon, for giving this presentation and the seminar, and we look forward to a discussion afterwards.
Kia ora koutou, and thank you very much, Dennis. That was really nice, and it's actually really nice to be back here at Waikato. And it was long ago enough now that 3/4 of my supervisory panel have, well, half of them have retired. One has left, but still, Ian Duggan, my primary supervisor, is still here. So yeah, it's really nice to be back and have the young gun Ian still around. And I've maintained connections with the university in the years since. So long may that last. So this is a talk which just covers some of the projects that I have been working on over the last few years. And I was trying to really print a theme out through this, which we can see in the title here, using stable isotopes to simplify food webs for applied freshwater management. But in the name of actually trying to be simplistic, sorry. Here we go. Sorry. This is what the title actually should be. So the more simplistic way of saying that is that stable isotopes, they do simplify food webs so that we can actually be using them in food web management. So just a quick run through. So I'm going to start off with, in a lot of ways, I'll be sort of teaching grandma how to suck eggs here. I know there's a lot of amazing ecologists at Waikato doing a lot of awesome food web research. But just quickly, what are we meaning by food webs? So firstly, we're talking about the community. So starting off with the community. So in an ecosystem, all the organisms that live in that place, that is the community. And the food web is the interactions that connect all those consumers within the food web. And it's one thing to look at the connections in themselves, but quite often there will be some connections that are more stronger than others and are exerting a stronger impact on this community. And a bit like when you've got lots of strings connecting in from different ways, is when you've got all these connections, that is what gives us some of these unintuitive, so pulling at one area can pull things in other areas in ways that we don't necessarily expect to see quite often. And when we zoom out and we look at some real world food webs, what we see is actually a lot of connections and they are really complicated. So firstly, that's the West Atlantic Ocean on the side closest to me. So I guess the way you guys are looking at it, that's the left, sorry. And then on the right is pretty small lake, Little Rock Lake in Wisconsin. So so just specifically focusing on that lake, that's a pretty small, simplistic ecosystem in a lot of ways, and there is a lot of connections there. And sometimes I think me personally, when I look at something like that, I can glaze over and think, where am I going to start with that? How do you even try to integrate all that complexity into some sort of a meaningful answer. And sometimes it can feel a bit easier to throw your hands up and say, well, it's complicated. We just need to be, and struggle to move beyond there in a way. But we really do need to actually consider food web interactions because it has real world consequences, particularly when we don't. So we're all in New Zealand. I think it's a bit of it's a national story, really, our failures when it comes to biocontrol, which such as introducing stoats to eat rabbits, which we all know that didn't really go too well. So we know that if we don't actually think about these trophic interactions and the fact that stoats might actually also eat other things that are a bit lazier and a bit easier to catch than rabbits, or a bit slower, I should say, then things can act, then there can be these unintended consequences. And similarly, one of the most famous examples globally would be the Four Pests campaign in China, 1960. So Chairman Mao declared sparrows as one of the 4 pests of the people of China, and then instigated a campaign to eradicate sparrows because of they were eating grain. So remove the sparrows, there'll be less grain removed, and therefore there'll be more grain production for people. What wasn't considered was the fact that the sparrows, they don't just eat grain, they also eat the locusts. And actually, so what ended up being an incredibly successful campaign at removing sparrows led to a big boom of locusts, a locust plague, and ended up being one of the biggest famines in modern history. And recently, well, a couple of years ago now, I should say, there was actually a court ruling that we need to be considering. We can't just say that it's too complicated and we don't know how to, we won't think about it, that we'll put that in another box. There's a court ruling that said, well, actually, we need to be explicitly considering these food web interactions as considered as part of the best possible science that we are driving into management. So this was in the context of fisheries, so Kinabaran, and setting the quota for rock lobster or quota, crayfish. And we know that crayfish, particularly up in the northern region, are the main predator of kina. And kina, when they get out of control, in high abundance, they will denude reefs of kelp forests, and then that has these wider ecosystem effects. So actually the quota for rock lobster now needs to be set so that it considers not just their reproductive ability and their sustainable yield, but also having enough biomass to be able to sustain a predation pressure on kina. So if we're needing to be using this knowledge and actually applying it, so how do we simplify all that complexity? And as I kind of alluded to earlier, I think one of the key messages for distilling some of that complexity is to follow the dominant process within a food web. So as an analogy, if we're thinking about our own diet, I eat potatoes and I also eat almonds. If there was a potato famine, my diet would be a lot more affected than if there was a shortage in almonds. They both fit into my diet, but one sustains me a lot more than the other and has a lot more of a meaningful impact on my personal population, so to speak. And a way that we can really get at that, I think, which a key message to take home if you're going to take notes at the moment, is using stable isotopes. So stable isotopes, I think, are a very good tool for distilling a lot of that complexity and actually allowing us to follow the dominant process. So stable isotopes, see if I can just get this. Sorry.
So if you back your
Oh yeah. Okay, cool. I was just trying to get the screen down. Okay, so the great thing is stable isotopes, they show connection and they show origin. So essentially, there's the old adage, you are what you eat. and you are. And so stable isotopes, that's literally the material that comes into your body is what builds your body, and it's what you excrete out. So you are the atoms that came from the environment that grew up and produced the food, and then when you pick that food, and then that is assimilated into your body, and that is essentially what we're tracking with using stable isotope signatures. And I like to just pause here for a little bit, and because one of the reasons why I was able to really embrace moving into stable isotope geochemistry was it came at a time when I was trying to connect with my taha Māori and connect with my whakapapa, and trying to understand a te ao Māori lens of viewing the world. And isotopes provided a great way of connecting that Western science, which I was doing my Bachelor of Science at the time, with that te ao Māori worldview, where that focuses really on that connection and the similarity, rather than at the time when I was trying to do taxonomy lessons and everything was focused really on differences. And so as a natural lumper, not a splitter, I found this as a really useful tool for not only in terms of science, but also in terms of being able to work between science and te ao Māori worldviews. And so yeah, I've just got two quotes up here that really kind of emphasize that. So one quote was from Te Wai Māori conference. So it is a lot of on the ground flax root practitioners. So explaining stable isotopes to a room like that can feel a bit daunting. And one person was just like, oh yeah, that's whakapapa, that's easy. And the other was Toby Curtis, the late chair of the Rotorua Te Aroha Lakes Trust. And I remember him saying about what makes Te Aroha, and I'm paraphrasing a bit here, but when you live on our lakes, you are one of us. And it is that showing that what it means to be Te Aroha, it means to be living on the lakes. So drinking the water from the lakes, and the lakes actually literally being part of you. Now, so just to work through what we're talking about here. So what is a stable isotope? So an isotope is just an atom that's a little bit heavier. And just in the same way that a heavy stone will move differently in a river, it will accumulate in different places than smaller, lighter stones will. Heavy atoms accumulate in low energy environments. So when we metabolize, typically we are breaking off the lighter, easier to metabolize atoms. So then the ones that remain in our body are those heavy ones. And what this looks like in terms, how we can use this in terms of a diet study. So I've got an example here with our cat, which is eating a mixture of birds and mice. And so typically how we measure this out, is so we've got our carbon on the x-axis, and that really shows us what our diet sources are. So we've got two sources that have quite distinct carbon signatures. And then our nitrogen values, they show us our trophic level. So what I was talking about with how the when we metabolize and we break down those lighter, we metabolize those lighter amino acids, those lighter proteins, and then we accumulate those heavier ones within our body. Because of that, we get this nice fractionation. So with each trophic step, you become enriched in nitrogen isotopes. So we can use this to calculate the trophic level of this cat. So we know that it's got a diet of 50% between birds and mice, and we can see it sits about halfway on the carbon signature between the birds and the mice here. And we can we can also look at its trophic level. So we can see that the mouse is eating the beetle, which is eating the grass. So the mouse sits at trophic level 2, and the bird sits at trophic level 1 because it's just eating the berries directly from the tree in this hypothetical example. And that's how we end up with the cat where it is. So now if the cat was to be a good cat, stop eating birds and just started purely eating the mice, we would see that in isotopic space, the cat would move, so it's sat basically directly above the mouse, so its diet would now be 100% mice, and it would move, it would be sitting so up in the nitrogen space, because it would be moving up to that third trophic level. So with a bit of an overview now, so so now that you're all done and dusted, bona fide isotope geochemists and food web ecologists, I'm going to start working through 3 examples of where we've been using, basically using to try and inform food web management within freshwater ecosystems. So looking at diet of longfin eels, looking at niche overlap for predator management, and then really looking at the role of littoral areas and for resilience within lakes. So starting up is looking at identifying these cryptic trophic connections. So as Dennis alluded to, this is the project that I'm looking at the role of inanga as a marine subsidy in driving the diet of longfin eels. So we all know our longfin eels are their populations have massively reduced, they're threatened, declining, and the Parliamentary Commissioner for the Environment 10 years ago released a report saying that they're on the pathway to extinction and there needs to be a moratorium on fishing. Now, the PCE at the time looked at all the drivers, and the risks associated with the eel population, their long life, slow growing, habitat degradation and damming. But one thing that wasn't mentioned once there is diet. And that's important because we talk about this being having a slow growth rate, which yes, they do, but their growth rates aren't inherently slow. We know that we've done work in the aquaculture industry to look at feeding up eels. We know that they can grow just as fast as any other eel species in the world, but they grow relatively slow compared to their potential, given their temperature within New Zealand. So in other words, they're food limited. That's what's limiting our growth rate of our longfin eels in New Zealand. So that suggests that diet is a major driver within why we see these such incredibly slow growth rates within our eels, and that's a major handbrake to conservation. The slower something grows, the harder it is to recover that species. And the PCE report, as you'll note in the yellow circle, it didn't actually once mention diet. So this is a management lever that we could be looking at that currently isn't being looked at. And to start with, we're looking at a case study of a ecosystem in the bottom of the South Island, so Waituna Lagoon, and Waituna Creek. So stepping back a bit, so we're fortunate to do quantitative electrofishing in Waituna Creek. As you can see, it's very agricultural stream. It's heavily impacted. It's highly channelized. It's basically being turned into a trapezoid culvert. The entire, most of the length of it, very little habitat, but it has got a lot of tuna in it. And so, it's got these pressures that were mentioned in the PCE report, but it still has a lot of tuna. And so what we're looking at is one of the drivers is, so it's a bit unique, Waituna Lagoon, in that most of the land use intensity is highest in the upper catchment and actually decreases down catchment until you get to a near pristine or one of the better lowland wetlands that we've actually got in New Zealand. So the Awarua wetlands, which you can, there's a picture of on the side here, is it's a Ramsar listed wetland, and it is and so there's the open water body component of that is the Waituna Lagoon. It's and the Waituna Lagoon has one of the last remaining populations of rupia, which is a macrophyte that can handle both brackish conditions and freshwater conditions. So it's a really important ecosystem engineer within this lowland system. And then a really important thing about the Waituna catchment is that, so we're talking about the creek having a lot of tuna in it, but also it's got a lot of galactid species, in particular inanga. So the inanga grow really well within this lagoon environment because it actually has these macrophyte beds, and we know that it supports high inanga productivity. So it gives us a bit of a snapshot in a way into one of the few remaining lowland ecosystems where we can actually go and have a look and understand, well, when we've got this lowlands ecosystem that's intact and we've got high production in these coastal brackish environments, and what is the production that we see coming up and how does that support our tuna population? So firstly, just looking at the biomass within the creek, just to give you a bit of an idea. So there's a national model that's set up for, it's a national scale model for looking at the biomass, calculating biomass in streams, and it's used for the National Stock Assessment. So that's the model estimates for biomass within Waituna Creek, and you can see the observed values. So it's about 400% more is the observed than the modeled values. And if we look at that on an international comparison, so it's an old paper, but Randall et al. 95, they did a global assessment of biodiversity of fish, biomass, I should say, of fish in streams. And what we see is that Waituna Creek, even on the international standard, is pretty high. So, there is a lot of tuna within the creek. So the next question is actually then to look at their diet and understand what is supporting this biomass. So just working through the isotope data again. So we've got the creek invertebrates on the left here, and on the right you've got marine, it's a lagoon and lagoon macroinvertebrate signature. So they're quite nicely separated. And this would be, so if we had a consumer that was eating a mixture of lagoon invertebrates and creek invertebrates, they'd fall somewhere along that line. The closer they were along that line to the left, the more their diet would be associated with creek invertebrates. And if they were eating the things that were eating the invertebrates, so if they were like a third trophic level, that would expect them to fall somewhere along the upper line. So just to go over again, so if the longfin eels that we sampled here were eating the food that's actually available within the creek, we'd expect them to fall within that circle. And this is where our longfin eels eat, where they sit. So these are all longfin eels that are collected in the creek, but they essentially look like very little of their diet is actually being driven, is being supported by the production within the creek. And in actual fact, a lot of their diet has been supported by the lagoon environment. What we see there is their diet becomes skewed more towards the lagoon invertebrates. We see that the trophic level goes up to being closer to or above that third trophic level consumer. So what is driving this? And what we see is so inanga here, and what we see is so inanga, they are eating and they're feeding in the lagoon environment, so they're assimilating that lagoon energy, those resources, and we know that they migrate upstream, and when they're migrating upstream, that is what is supporting our longfin eel populations. So as an analogy here would be thinking of our inanga as salmon, and if we think of our tuna as bears, and if we were to transport this as a model system over to the Pacific Northwest coast, where we're over in the US on that Pacific coast there, the diet of bears is incredibly similar actually to the diet of orca whale, even though they're miles apart. Their diets are functionally incredibly similar, and that is because of the role of these of the salmon in transporting these resources miles up catchment. In a similar way, we've got an analogy here in New Zealand where the inanga are transporting these lowland or these brackish coastal resources up into our catchments where they're then being consumed by longfin eels. So here the diet of longfin eels, it was 75% of their diet was supported by was subsidized by the lagoon-derived inanga. So that's a substantial portion of their diet. So in terms of looking at this, how important is this in terms of growth? We've got, it's really good to actually look at we can use the natural variation within the system as a bit of a natural experiment to try and tease that apart. So one of the great things about the lagoon is the inanga, their life cycle goes out into the marine environment and then back into this lagoon environment. So, and this is sorry, I should have mentioned earlier, this is a barrier beach lagoon. So sometimes there is open access to the ocean, and sometimes after a storm, the beach the beach will get closed off, so there's no open water access, and at that point it becomes essentially a freshwater body or closer to a freshwater body. But when that happens, the inanga cannot complete their life cycle. So in years when the lagoon is closed during this migration period, we get essentially zero recruitment of inanga. And so the longer the open period, the greater the recruitment that we get into the lagoon. And we know that once they get into the lagoon, and they've got a fantastic feeding environment, then we get this really high productivity happening. So we're fortunate enough to harvest 7 tuna to actually quantify the growth rates looking at the otoliths. So counting the growth rings within an otolith in the same way that you would count the growth rings within a tree trunk. So these eels have to be harvested. So we're working alongside Te Awarua Runaka, and so the eels were harvested. They were taken for kai, but the heads we took and we extracted the otoliths from. And then we were able to take the age, and then knowing the lengths and weights, we can then back calculate what the actual growth rate is. So these are the 7 eels that were harvested, and we can see the growth rate here in millimeters per year of these eels. So there's a bit of variation, but if we compare them to the average of tuna longfin eels in Southland, they're all growing a lot faster than we'd expect to see, than what we do see, I should say, in Southland. And what I've done here is, so I mentioned earlier about the eels growing slower than we'd expect them to. So essentially we've got a theoretical range based on the temperature data within the stream, what we'd expect to be the maximum growth rate of eels, and also expect essentially to be the minimum growth rate of eels. So that's from a bioenergetic model. What we see is that, they're getting up towards that around 50% of their maximum potential growth rate. So just going back to now looking, well, I was talking about the lagoon open days. So if the lagoon's closed and there's less fish available, we'd expect to see that come through in lower growth rates. And that is exactly what we see. So years where the lagoon was closed off and we couldn't, and there was no access to inanga into the system, the longfin eels, they grow slower in that year, as we can see with smaller annular increments within the otoliths. Now, first thing you'll probably note is that there's actually a lot of spread in that data, and that's because the data is actually masking the individual effects of individual tuna, and that varied significantly. So we see some eels were incredibly responsive. So when the lagoon was open, their diet, they seemed to switch their diet very quickly to target those inanga, and they grew really fast. And there was one tuna number 5, which was the opposite. It seemed to actually have a negative relationship with lagoon open period. Now, if we actually look at that that response relationship, so essentially the slope of that line, what we see is that the tuna that are responding, that are very responsive to the lagoon opening regime, they're the ones that are growing fastest, and our one that's got that negative relationship is the one that has the lowest growth rate. So put this together, we're basically seeing that this diet subsidy is driving a growth response. And furthermore, just looking at the what we see is just looking at the isotope, just bringing this back to the isotope data. So those tuna that have the that are the most responsive are typically the ones that are also have the highest proportion of lagoon diet, with the exception of one outlier. But the time that we were sampling the stream was when the lagoon had actually been closed off for quite a long period of time. So we think that there was a bit of a mismatch where these really sensitive, rapidly responding species, or individuals, I should say, individual was actually probably switching diet to the rather anomalous situation of the lagoon being closed for 2 years. So just to put this into context, so a rewilding approach to conservation, so essentially focusing on diet or restoring food producing habitat as a way of as a pathway forward for conservation, particularly for our long-fin eels. And so one of the important things, so if we're thinking about this in terms of what is the big, what is it that's driving this within the Waituna Lagoon, it really is the fact that we've got this unprecedented lowland wetland complex that is able to support the production of these inanga. And we know that wetlands are incredibly productive for fish. So the global average is 800 kgs per hectare. We know we've lost a lot of wetlands within the country. So, in total, we've lost 2. So, if we were to essentially extrapolate that out by the amount of lowland wetlands that have been drained and converted largely into agricultural land, or in some cases residential land, that would equate to about 2 megatons of fish production that we have lost per year. So without trying to focus too much just on the doom and gloom, actually looking at some of the potential positives of this. So we actually went out and we looked at inanga biomass in streams that have had some sort of constructed wetland placed within the catchment versus controls that are just your typical, these are urban streams. So 6 urban streams with some constructed wetland in the catchment versus ones that have no constructed wetlands. They're just a typical stream. Firstly, I'd like to add that these aren't wetlands that are placed in there to enhance fish. These were wetlands that were put there mainly actually as storm retention or some sort of a flood conveyance in mind. So slowing flows down basically. But in all cases, these wetlands, although it was unintended, they all boosted the biomass of inanga. So that's the first stage, is that we're showing that there's higher biomass. We're wanting to actually look at compare the growth rates of the inanga within these streams. So actually look at how these wetlands contribute to the annual production of inanga within the system. But there is as we're moving in, so we've got the Kunming protocol, Kunming Montreal biodiversity framework. We have tentatively agreed that we're going to restore 30% of our degraded habitat by 30% by 2030. It's quite an ambitious goal, but I think there is a real opportunity here. We know that we've lost a lot of wetlands, and we know that we can have this huge, big leveraging effect of restoring wetlands for our wider freshwater ecosystems. So I'm hoping that can actually be a bit of a call to action in a lot of ways. Just repeating that point there. So there's a lot of wider ecosystem benefits for these for wetland restoration. So moving on to the second topic, which is linking theory and application to guide management. So this is a bit closer to home. This is working down in the Te Arawa Te Arawa Rohi, and particularly we're looking at Lake Waikato here. So, so Lake Waikato, it's one of the it's a near pristine lake. It's the most pristine lake within the Te Arawa area. There's one road in, one road out. There's no housing around the lake, and generally it's got good water quality. And there's a lack of introduced of invasive species in the lake, and it's actually manageable with the one access point. We can maintain it as a near pristine lake. And one of the ideas that has been considered is, well, what can we be removing, scaling back some of the introduced species that are in the lake to be making it essentially a wild lake ecosystem. And with in particular focus on trout, which are currently stocked, rainbow trout, which are stocked within the lake at the moment. Now, so if we were to, so this was started with the situation of thinking about destocking trout to the point where there is, we were not to have trout within the lake. So thinking about that, so we think about what the pre-colonization state of the lake was. So the lake was filled with inanga. So inanga were in the lake, they were highly abundant, and they were a major food source. And in the turn of the century, rainbow trout were introduced into the lake. They predated on the koaro that were, sorry, I said inanga. I meant to say koaro, sorry. They predated on the koaro in the lake. And then to the point where they drive that population right down. And then the Climatization Society at the time then added common smelt brought from the Waikato River on the train over to the Rotorua Lakes and spread throughout the lakes as a forage fish to support the trout fishery. Smelt, they grow, they're an annual species typically, they grow really fast, and so they can, in the pelagic, they really support trout fisheries. So if we were to remove, because of that, if we were to remove trout now, we're not actually going back, we're going to a third state, a new state, which is where we've got smelt and koaro trying to coexist. But this is actually a different scenario, and it's something that we don't see very commonly. So we're actually trying to put some tools together to understand this. And so we don't want to be going into a situation where we were to remove trout and then look at the consequence. We're wanting to see what tools we can actually develop to try and inform that management better into the future. So this is part of a wider project with the Fish Futures program. We're actually developed multiple lines of evidence approach with modelling, spatial distribution, and some of the field data that I'm presenting here. But I'm just going to focus on the food web data here at the moment, and because we have little understanding of what that state is. So what we can actually do is we can look at the theoretical understanding of also what does this mean. So if we've got just koaro and smelt cohabiting, or coexisting within that environment, we expect that there might be competition, and competition can often lead, if it's severe enough, can lead to the extinction of one species. So we can actually put this within a framework. So there's a Locked-in Altera competition framework, which we can actually put this data into to see what we might expect the outcomes to be. So if we think about so competition being niche overlap, in this case we're thinking of diet. If the two species have completely separate diets, they don't compete, so they're just sort of cohabiting within the environment, and that's completely fine. As their diet starts to overlap, we start getting competition, and then we get different stages of competition. So if the competition is at these intermediate levels here, then we expect that essentially within their Locked-in Altera model, that basically the same outcome will always essentially happen. So it's a stable state. So there's one solution to this problem, which is always the same, no matter, regardless of the starting conditions. But if the competition starts becoming extremely severe, and the overlap of diet overlap is really strong, that actually gets to the point where maybe where you can disturb the starting environment, you can change those initial conditions to the point where you can actually pick a winner of that competitive interaction. And that's where management options might be possible. Just a little bit of a shout out here, just back to Waikato days, because at least when I was here, Brendan Hicks was taking a class in population ecology, and actually one of the modules was actually looking at Locta Volterra competition, and it was like, that's kind of neat. It's pretty theoretical, but it's actually quite cool that it can be applied into real world situations. So here you go. So just looking through, so what we see is that, so firstly, this is looking at the pelagic diet and how in the size structuring. So what we see with quota and smelters, essentially, they do fill pretty much the same niche. So their diet is largely pelagic, and then as they grow, their diet becomes less pelagic. So that's feeding in the middle of the lake, and they start feeding more on the sides of the lake. So very similar functional role, but if we actually look at the diet overlap at any given time, what we see is that we've got that essentially that intermediate level of niche overlap. So if we were to actually put this into Locta Volterra framework, what we see is we've got stable competition. So it's very unlikely that having management interventions on smelt, so doing any some kind of a smelt disturbance, so to speak, would manage to flip that interaction. And when we put this together with the wider literature, what it suggests is that basically, and what is going forward is going to be a very a slow and precautionary approach to reducing stock numbers within that lake to avoid any major impacts on the quail population within the lake. So third example, and maybe people recognize the boat there. That is, so this is actually work from my PhD when I was here several years ago on Lake Taupo. So the work was really looking at understanding how the water quality within the lake is driving the ultimately getting up to supporting the fishery. And so if we're trying to if we're looking at this and we're looking at, again, trying to focus on these dominant players, what we see is the biomass within the lake at any given time is typically dominated by smelt. So smelt are these pelagic fish. Lake Taupo is this big, large, open pelagic bowl, and they so what we see is that that they really can use that pelagic open water environment better than most other fish, and they really dominate the biomass. So essentially, what happens to smelt is really impacts what happens to the trout fishery within the lake, and smelt really act as a conduit between the productivity or the phytoplankton at the base of the food web, and how nutrient management might impact that, and then as a way of sort of funneling that effect up to the trout fishery at the top. Now, if we're looking at so forth, so we see there's that really strong driver of pelagic production. We see smelt are these pelagic fish, and then they support the trout. And so even if we just we created a model just looking at a measure of a rough measure of pelagic production. So we know that in Lake Taupo, the duration of the winter mixing, so that's just a very physical process, controls the amount of annual production. And so just a simple model looking at with the one driver being the amount of annual production, the amount of mixing days as a driver of that smelt population, smelt, and then we can actually do a really good job, pretty good job, I should say, of recreating historical trout biomass over long periods of time. But yeah, as I say, it's does reasonably well. But one of the problems when we think about that pelagic production is that it's hugely variable within the year. So on orders of magnitude, it goes from, when the lake is mixing and there's the nutrients available and there's this boom in productivity, which might last 2 to 3 months. And then the availability of those resources just crash. So what's happening for the rest of that year? If we've got this huge biomass of fish, which is presumably out there growing on these resources, when these resources crash, it's really, in which we know predictably that they'll do each year, what does that actually mean for the wider food web? So this is seasonal sampling of smelt, the green bar on the bottom, and common bully in the orange in the upper bar. So essentially, a little bit unintuitive. What we're looking at here is the littoral diet contribution, so the opposite of pelagic diet, which I've been talking about. So what we see is that, after that winter mixing period, we see that the smelt have a very low contribution of littoral diet, which is what we expect because they're feeding in the pelagic zone. And overlaid on top of that is an average abundance of zooplankton, their main diet, within over that same period of time. So we see the zooplankton abundance decreases through time because the low productivity in the pelagic environment, and the diet of smelt goes up to being over almost 70% supported by littoral habitat. If we look at topor, as I said before, it's essentially a big bowl. There's the littoral habitat is just that tiny ribbon of getting into the purple zone around the lake. So that's the area where down to around about 40 meters depth. So it's a tiny little habitat of the lake. In fact, it's less than 10% of the lake, and at times of the year, 70% of the animal biomass is actually being supported by 10% of the lake area. So if we're thinking about the resilience of the lake and actually managing it, we shouldn't be thinking just about the pelagic productivity, but also the littoral productivity, and actually managing that those two factors together. And actually, if we think back through time, so we look at our model at historically over predicted the abundance of trout, and in later years it actually under predicted trout abundance. And if we think about that, what we see is that when we've got more nutrient, when we've got those higher nitrogen loads going into the lake, we expect to see higher pelagic productivity, but just again during that that peak period. So during those periods, we might have more growth, more production of smelt, but then that's still inevitably all going to drop out. And then there's this higher biomass of fish that's supported, that needs to then be supported by that same limiting resource of littoral habitat around the lake. And this is important because at the moment we don't really do a fantastic job of managing our littoral zones within lakes, but we should be. This is where we're actually, most people actually interact with lakes in that area. Now the first point of contact where our land use pressures come in, and also they're quite physically dynamic. This is waves crash largely on the shore. That's where that energy is all absorbed, and it's where we build things like breakwalls and jetties, and normally remove physical structure like logs that might be in the way and things like that. But what we know is that they're also very sensitive, and they are often the first places to be impacted by eutrophication. So this was some work we're looking at. So benthic filamentous algal blooms, so occurring within lakes that are otherwise got good water quality. So we might be offshore measuring the water quality within the lakes, and the lake remains clear. It's still within essentially, pristine, good water quality within an A band if we're talking in the New Zealand water quality framework. but near shore, you might be having these problems that is being completely missed. So, if we're actually wanting to be understanding the lakes, moving beyond water quality and focusing on these wider food webs, we need to really be focusing on the littoral zone as well. So there's some examples of other places around the world where other lakes around the world where this is happening. So some of our larger, more iconic lakes. So we're talking about Lake Tahoe in Western US, and then also Lake Waikato in Siberia, where both pristine, very low nutrient, clear water lakes, very iconic lakes, but where these impacts are being felt within the near shore zone, while the open water quality remains stable over time. So finally, looking ahead, I've presented 3 examples that are quite intensive on 3 relatively well-studied systems, but we've got over nearly 4,000 lakes within the country that we need to be also, we're wanting to be doing the similar management for. So how can we get food web data for the many lakes that have few data points at the moment? So this is a project that working with crew here on the OLAF program. In fact, actually, Frank Burden and myself will be co-supervising a student on this project, which is essentially trying to develop a meta-web framework, so understanding the known trophic interaction so that we can then build food webs based on the presence of species in order to try and use, so we can then integrate that with eDNA data, which is scaling up at a huge rate and allows people to sample lakes on a much broader scale and much more cost effectively. So we can actually be trying to develop food web understanding for a wider range of lakes. So this is just a next step into the future, which we will be looking at, and so something that I'm quite excited about, and so watch the space for it, essentially. So just to round up, food webs, yep, we do know that they're complicated, but also, yeah, they're doable. I think that we actually have the tools available to help us be integrating some of this food web knowledge, and we can actually be using it very practically. It's sometimes I think that food webs are thought of as this very esoteric theoretical thing, when actually, this is very practical information that we could be and should be using within management for better outcomes. And I say for freshwater here, but I think that actually applies wider. So yeah, thank you very much.
Thank you very much, Simon. And that was quite intriguing. So we have a few minutes for questions. Please, if you have any questions. Sure.
Yeah, Frank.
So I've got quite a few questions, but one I'll see more put it all later. I was interested about Lake Taupo because the littoral zone is obviously quite important 7 times of the year. But I was interested if there's any knowledge known about terrestrial inputs. So I know Bob Dow's talked a lot about the chafer beetle when they hatch and they sustain the trout fishery in Lake Taupo. But I was just wondering if you had any sense about that and whether that drives like apparent competition or rate switching that may have some consequences for the food web.
It's a really good question. I like in terms of trying to detect a terrestrial signature, I think that would be the littoral, because you've got essentially carbon limitation within the littoral zone because it's stuff that's attached down low. It's got quite a distinct signature from anything that's happening in the terrestrial zone. So we didn't pick up. We would have picked up that terrestrial signature, and we didn't. But in saying that, I suspect that those, this was a 1-year study, and I suspect that those are sort of, decadal, multi-year cycles, thinking like masting events and stuff like that. Yeah, and it would be interesting to actually look at that, and so slightly unrelated, not quite necessarily topal, but thinking of those terrestrial signatures, there is, we had a summer student that was working with Ngāti Kitirato, so that's the Nelson Lakes area, and looking at tuna otoliths up there, and the growth bands, the annular growth bands, they could actually relate, because these are very old, long, you're looking at sort of 80-year time scales, and they could actually see they could actually detect the masting events within the growth bands. So those signatures can
inputs or something, so mice are up to go hard on them, but I'm sure the big tuna will eating mice. Cool. Yeah, nice.
Maybe time for one more question.
Can you walk through the process of you concluding that moving trout would lead to the smell taking over even within a stable condition?
So essentially the competition is enough that that they don't coexist, and we know that from just looking at spatial data. There's no records of lakes where smelt and quota coexist in the absence of trout, and we know their diet overlap is enough and we know that smelt grow faster, that basically they're always going to outcompete them in essence.
In the absence of predation.
But they are much more susceptible to predation, and their populations get cropped, and so that cropping sort of maintains that coexistence.
If we're taking Simon out for lunch after this, so if you guys, there's an open invitation for people who want to come along for further chats on food web dynamics in lakes, or much broader than that, please come along. And please join me again in thanking Simon to come up to Waikato University and give this seminar. Thank you. Small token for appreciation, actually, a little tonga for as a reminder that you've given this seminar. So thanks.
Got it. Thank you, everyone. Yes.